So yeah, let's say I believe there will be very last minute folks joining us. But hello everyone, welcome to ASCA Seminar Series. This is John, the seminar coordinator. So I will be moderating the seminar for you today. Just so you know that this seminar is being recorded and we will post the recording on our YouTube channel. Coming up, we will have like two more in-person seminars and a bunch of virtual seminars. So please stay tuned with us. And before the speaker get started, our director, Lars Peter, also joined us. So I'm going to pass the ball to Lars Peter. So Lars, please go ahead.
Thank you very much, John. And it's a pleasure to welcome and introduce Michael Gooseff, professor and associate dean for research in the College of Engineering and Applied Science at the University of Colorado in Boulder. And as far as I can gauge from your CV, you have been at CU your entire career, basically.
I moved around a little bit, but I did
a little bit. But okay, so you're back to your alma mater. It's a better way to put it. You got your education up through your PhD at CU. You moved around, and you're back. Yep. And you have a long history of studying the dry valleys around McMurdo, and that's what you're going to be talking to us about today. So many people may not realize that, but East Antarctica is actually a desert. I mean, we think of it as a huge ice cap, and it is, but it's an ice cap because it is extremely cold there, not because it snows a lot. It's dry. But even by those standards, the McMurdo Dry Valleys are drier than the rest of it. And you're going to talk to us about, I assume, how we still find life there. I mean, what is actually behind that? So this is a new topic, I think, for many of us. So we look forward to hearing what you have to say. Over to you.
Terrific. Well, thank you so much. Thank you for inviting me. This has been a lovely exchange, and I'm looking forward to questions now. Let me see. Let me get into that mode. And you're seeing the presentation mode. Is that right? Someone just give me a
yes.
Okay, terrific. All right, I have both on my screen, so just want to make sure you're seeing the right one. Okay. Okay, what is? Well, this is interesting. Okay, there we go. So yeah, I had the distinct pleasure at the long time ago of getting the offer to go to Antarctica to be on the stream team, that is to study streams in the dry valleys as a PhD student. And it was phenomenal. And so after working down there for a couple of years, I knew I had to find a way when I became an academic to write new proposals to go down. So I had a couple of other projects. And then one of the things that I got the real honor to do is to become a co-PI of this long-standing research grant. I'll give you a little bit of history and context of the LTER in a moment, but this is a view of Lower Taylor Valley, and so we often see these cold-based glaciers on the right. That's Canada Glacier, and it abuts Lake Frixell there on the left. We'll go over a map here in a moment, but just to give you a sense, this is kind of springtime, so maybe October, November, and this distribution of snow is actually quite common. We don't get blankets of snow there. When it snows in the winter, there's usually a lot of wind involved, and so a lot of the snow gets patchy. It builds up in topographic depressions and so forth. Now, I had ecosystem on the slide on the title, and I wanted to sort of honor that because the LTER project, the E is long-term ecological research, and ecosystems really depend on these connections that I'll be really talking about more than the communities in the rest of this talk today. And they count on these connections, whether we're moving heat or matter or organisms. And we'll talk a little bit about some examples of several of those. These vectors facilitate resilience, which is to say that if there is some kind of big catastrophic event, some disturbance to the system, they facilitate being able to respond to that, to have these communities, biological communities, come out on the other end. And it's really about connections through space and time. So I'll touch on a little bit of both space and time. And one of the great opportunities that we have is because this project started overall in 1993, we have a wealth of knowledge that we've been collecting since then to really think about that temporal component. All right, here are the folks I might be misrepresenting. From left to right, these are a bunch of the current and former PIs on the project. And these colors don't mean anything except they were better contrast for the background at each spot here. But I just note that we have 5 new co-PIs as of the beginning of our current incarnation. We're just starting year 3 of the 6th incarnation of this grant. And we're funded on 6-year cycles, and it's been just a real pleasure to work with all these folks. But I'll do my best to represent them appropriately. I'll just note that when we go down to the field, even though we are one big team, we get divided up into these field teams. And so you can see each one of these has a theme to it. And that's helpful for the U.S. Antarctic Program contractor to kind of figure out how to support us and so forth. But it also is helpful to us then to think a little bit about what are students doing, on this field team or that field team, and how are we working across these teams and making sure that we can kind of work out our logistics and our field plans. LTER is actually a network. So the National Science Foundation funds about, I think we're at 26, 27 of these sites right now. But one of the interesting things about the LTER model is that you're expected to do cutting edge ecological research, the science component, but you really have a mandate to monitor your site as well. And that's sort of unique at NSF. The monitoring is part and parcel with the science that's going on, and those long-term records then get published. You can go to the EDI, the Environmental Data Initiative, and find almost any LTER data set that's been collected over the past 45 years or so there. We are funded on 6-year grants, as I noted before, and that's a renewal process. So we're not necessarily going in and competing with each other on those renewal cycles, those 6-year cycles. And this provides the opportunity for short and long-term perspectives. So the long-term perspectives are big field experiments that you could run for a decade or longer. The short term is, well, most students are not in graduate school for a decade. And so, we can also think about ways that they can come up with a measurable sort of piece of research, of science that can inform the broader goals that we have in our project as they were written, even if they are not all leveraging just the long-term experiments or data. So there's some mix of that occurs on sort of short time scales. And because of this network, we come together every 3 years, or at least we have until recently, to synthesize. So to bring our results together and sort of think about how things are working at many different locations. So hardwood forests, as well as our polar desert here in the Dry Valleys, for example. So 26 sites across a lot of different ecosystems. That includes marine. 45 years of existence, over 7,000 data sets, and 27,000 plus publications. And really, it's a network of thousands. When we think about each one of these 26 sites and the folks that have been working at them over 45 years, not every site is that old, but all the students and technicians and PIs, there really are a lot of folks that have been involved with this network for a long time. Now, one of the things that makes us what I would consider to be a competent network is that we're all each mandated to measure these 5 things. And these are all very ecological, but I want to there's an important reason that I'm bringing this up. So I'll come back to that here in a second. But one is primary productivity, right? How does photosynthesis occur in your site? And maybe that's different for a grassland than for a polar desert, but we all are still trying to kind of figure out what rates and what is that fundamental underpinning of ecosystems doing. Nutrients, how are nitrogen and phosphorus, kind of the 2 big ones, being cycled and transported in your system? How does your system respond to disturbance? What are the typical disturbance types? We're not going to have forest fire impacts down here in the Dry Valleys, or at least not direct ones necessarily, but we are, and we probably won't see, maybe we had a flood year, particularly warm year, and our colleagues in Moorea who study the coastal ocean may not see a similar type of warm event that causes a lot of stream flow. Like our sensitivity to warmth is different than theirs. Organic carbon, how is it generated? How is it cycled? How is it transported? And then finally, thinking about biological populations. How do the populations exist? How do they thrive? How do they interact with each other? And so what's wonderful about this is all of us can bring our findings from these 5 core areas to these all scientist meetings and really develop new theory more readily because we're all doing this in these, making these measurements in different locations. But a number of these also are dependent on these connections that I'll talk about for the rest of this talk. Okay, so quick overview of the landscape. We have soils, glaciers, streams, and lakes. These lakes, by the way, are permanently ice covered, but they do have these edges, we call them moats, that thaw out every year, melt out, and so there is liquid water there. So the picture you see behind me, in fact, is standing on a shoreline looking at the liquid moat and then the ice cover in the middle of the lake. And these moats are really fascinating places. I mean, they're dangerous in the context of, do you walk out on thin ice or not? And, you don't want to fall through. But they're also fascinating because the ice isn't in the way of sunlight getting through and so forth. And so they're really productive areas with lots of algae. Now, we have a conceptual model of our landscape that looks something like this. And we think about this as what we call the low energy input, or a season that may not be super warm, or maybe it's cloudy. There's a reduction in the amount of sort of energy that's coming in at the surface. And in that case, we have, sort of a system like this. We still have some meltwater inflows. We still have the moats that thaw out. And on the right-hand side, I'm going to modify katabatic wind. That's how we used to think about this. But now we actually know that these are foehn winds. So foehns instead of katabatics is different driver, but we still have big winds that blow down these valleys from the polar plateau. And that's an important trigger for vector for moving material around. And then we compare this now to a high surface energy input system season, summer, austral summer, where there's much more meltwater inflow. The moats might get larger. The ice on the lake may shrink a little bit, and it may not be as thick. It may thin a little bit. And we may still have these foehn events, and we'll talk about the details of those here in a second. But we don't always know exactly what kind of a season we're going to have. Is it going to be high surface energy or low surface energy as we get down there? But we're trying to make sure that we're prepared for both in our experiments and our normal operations. All right, let's start in the top left here. We're at about 77 degrees south. So if you look at that red, if you can see my cursor or arrow, we've got this red pointer right here, a red box. That's approximately the location of the dry valleys. At the scale of a couple 100 kilometers here, we are now looking at these main valleys, and 4 of them then are sort of called out. And so this was a figure for one of our proposals where we tried to identify, this is where we're doing, work, where we have, monitoring side of one kind or another. I just want to point out Taylor Valley. We're going to talk about Lake Bonny. Lake Bonny has a west lobe and an east lobe. So there's like this sill kind of dividing them under the surface of the lake. Lake Hoare exists right here, and Lake Frixell here closer to the mouth. So this is on the far right of this particular image for Taylor Valley. That is the coast. And so the McMurdo Sound starts right there with that ice. And we're going to talk about a number of these lakes and so forth, and I'll go into some detail, but I just wanted to give you a little bit of an overview of this particular site. And we do have 4 fixed camps, well, 5 actually, within Taylor Valley, and those provide really the basis that we work out of when we go out into the field. Life is small in this ecosystem. So I showed you the view earlier. One of the reasons that life is small is because large animals cannot really exist that well. We see a number of these mummified seals across the valleys. They've wandered inland and they've kind of run out of sardines and whatever else they've eaten and have passed. And so they get weathered by the wind and the sun and the cold through time, and they're kind of a stark reminder of what this place is like. And so on one level you say, well, this is a harsh environment, and it is harsh for the animals that don't have the resources here, and that includes humans to a large part. We import all of our food and so forth. But in any event, life has to be kind of small in this ecosystem because there really just aren't enough resources to work their way up to larger organisms. In the lakes, we see a lot of benthic algal mats, particularly as I mentioned earlier in the moat regions. When those mats detach, they'll float up to the bottom of the ice cover and get incorporated in and work their way up. So we see that this carbon that's generated, this biomass that's generated at the floor, the bed of the lakes will work its way up into the ice cover and work its way up through that. Now, the ice, by the way, melts or sublimates off the top, and then new ice is added from the bottom. So it's kind of an upward conveyor belt. So when this algal material gets stuck on the bottom and works its way up through that sort of elevator up through the lake ice, it eventually can get out and be blown around the valleys. So interesting sort of transport there. In the streams have these beautiful shag carpet kind of algal mats. And so this picture on the left is a stream where the black along the edges this is a nostoc, mostly mat, and we typically find those on the edges of streams. And in the middle of the streams, we see more of this orange formidium dominated mat. And then if you go into little crevices, you can find some green material and so forth. So pretty interesting. And now these just dry and desiccate through the winter. And then in the summer, they come back when stream flow starts. These are also places that harbor an awful lot of a huge diversity of diatoms. And we'll talk about how we've used and studied diatoms a little bit here in a few slides. In the glaciers, we actually have life up there too. A lot of this material that has either come from the streams or the lakes has been blown up onto the glaciers and sits within, sort of melts its way in and then creates these cryogonite holes. And so these cryogonite holes have actually been measured to go through metabolic processes. So they are breathing more or less. They are creating oxygen and consuming carbon. And consuming carbon in these little icy pots kind of across the glaciers. So pretty fascinating spots as well. This is just a little clip that I got from Ariel Waldman. I'll just note her webpage here. She's a filmmaker that came down and has worked with us a couple of different seasons. And this is a view under a microscope. So these are, you can see a tardigrade moving around over here. You can see a nematode. And this is the microscopic life that is abundant in this valley and does not find this valley, this location and climate particularly harsh. And we'll talk a little bit about how some of these small guys do get moved around in the winds that we have. I'll come back to Ariel at the end too and give you some more information about how to find out some of the work she's doing. Okay, so let's start with the winds. What about this transport that's going on as we have high winds through the valleys? Now, the original paper, this Nyland et al. 2004, we were still calling them catabatics. So I've tried to correct that language through here. But you'll notice that from the records we had at the time, getting into about 2003 or so, we had about 10 years of data. You can see that what we qualify as a phone, so that's based on wind direction and intensity, or wind speed, and the fact that we'll find that for a prolonged period of time, we see those conditions generally occur in the austral winter. So June, July have really high frequencies, and the wind speeds overall, in other words, just thinking about the monthly average wind speed, which I realize is a bit chaotic to try to average, but the average wind speeds tend to be highest, in the winter and then into the coming spring as well, into September. What's really fascinating about each one of those phone events is you can see two different plots on each one of these three graphs. And the first one is Lake Bonny as the solid data. So these are from the Met station at Lake Bonny. And then the other one is at Lake Frixell, the dashed one. And you can see this very persistent, we go from sort of 1 to 2 meters per second, sort of bouncing around. And then we have this big jump, right? On 5/28, May 28th, we see this big jump here. We stay at high wind speeds for a few days. And at Frixell, it sort of ramps up a little bit later and then comes down. And at the same time, relative humidity plummets. So the air gets dried out essentially. We see that happen here at Frixel as well. And the temperature goes up. So we go from almost minus 40 to about -5, -3. So it's not uncommon for that to happen, that departure to occur in the winter, which is a little bit baffling. I mean, I think we think of just -40 out there, but the fact that this can move up and down and have such striking differences and changes is really speaks to the dynamic of this system. Now, we're looking at this big rock, and I was surprised to walk across this and find all of this fine material collected. So we're looking north, and up valleys to our left in this picture and down valleys to the right. And I'll just highlight for you, this is the collection of material behind this rock. Now, I expect we've all seen something like this before, but it's particularly prominent in the dry valleys. And maybe it's because there just hasn't been a lot of people walking around and tromping on it and so forth. But this is a place where that material that's been blown across the valley has been captured, at least some of it, right, in some of these locations. So we tried to make use of some of these and say, okay, well, what's been blowing around, right, and where, and who's there from an organismal perspective. Now, we also will find sort of what I might call evidence of a lot of wind transport on the lakes, the lake ice cover. So this is an ice cover from, one particular early summer, probably November or so. And this is from another year where you can see a lot of material is collected on the lake ice, and that will try to melt its way through, and high concentrations can actually do that to sort of defy the elevator that I talked about earlier. And then we have sediment traps in some of these lakes to capture what comes through and see what's in it and who has fallen through and so forth. Now, when you get down to, it's one thing to take a picture from a helicopter, and like a lot of tundra that you might see pictures of from afar, it looks green and rolling and so forth, but you start walking across tundra and you will roll your ankle pretty quickly, right? There's all these tufts out there, and that's not unlike our lake ice here. So we see that the sediment, where it collects a lot, melts differentially down into the ice, and we get these towers of ice, and there's actually quite a bit of meltwater in there, but it's not that deep. It's about as deep as your knee. And so, when you bust through, it's not like you're going to drown, but you will probably bust up your knee and maybe get a cut and so forth as you're walking through this. So it can be quite challenging. We've all tried to figure out our own way of predicting where the good and bad ice is to try to move through these. This is an average size. I realize this perspective might be a little bit off, but this is on Taylor Glacier, which is a particularly smooth glacier. This is one large concentration of material, and we're quite a ways from either wall on the side of the glaciers here. And this is a graduate student of mine. He's actually taller than me, so this is quite a big collection. But this is another one of these sort of aeolian deposition locations that we could sample to try to understand what's moving. And so I've had a couple of attempts of doing that. So Marie Sabaka was a PhD student with John Priscu back in the 2000s, and one of the things that she did was and these were data that were collected before her dissertation. So another example of something we were doing long term, but she was able to harvest these and work on them. You can see that the aeolian flux on the left y-axis are the bars here, the gray bars, and you can see that on the right y-axis are the phone frequency. So how often were these phones occurring? And when we just look at the three highest phone frequency years, we also see the highest aeolian flux for those particular years. So on the order of a kilogram or so of per meter squared of sediment per meter squared per year. And then she went in and interrogated those and tried to figure out, from a chemical perspective, what kind of nutrients are in there. So I'm just going to show you the particulate data here on the next slide, but PC, so P is particulate, so PC is particulate carbon, PN is particulate nitrogen, and then PP is particulate phosphorus. And so these are at least the building blocks of ecosystems and organisms we know of. And then she stratified these across 5 locations that she could sample. So one is just aeolian captured material. We have these sort of aeolian collectors at different heights. L is lake sediment, and so that's going out onto the lake and finding that sediment, sampling it. G is glacier sediment, which are sort of more superficial than C, which are the kryogenite sediment. So that's where we've got a depression that has been generated and melted its way in. And then S is for soil. So what does the soil look like? In just in general. And so, when we look at carbon, there's cryogenite sediments have higher particulate carbon, nitrogen, and phosphorus compared to all the other locations that were sampled. And maybe that should make sense because there's also life going on inside of those cryogenite holes, right? They are processing the nutrients that come in, and there might be enough for those to sort of be picked up in other ways. But the other thing that I think that's kind of interesting here, though, is we look at the aeolian capture is pretty low. I mean, what we were able to sort of catch just moving through the wind was not that high compared to places where it's been collecting for some time, which maybe speaks to the temporal component of this long-term aeolian flux. All right. The wind also transports life. And so these are microscopic organisms that we often find in the soil. And we have nematodes, tardigrades, rotifers. Sometimes we find some mites as well. And so all of those are moving around during these high winds. But because they're so small, we don't always see them exactly, right? So if we just look at we've got live and dead here. So just noting, in general, the organisms we find, in our soil and lake, soil to lake glacier transport. So in other words, sampling those places on the lakes and the glaciers that have sediments collected, we find, about 70 individuals per kilogram of sediment. And then, about 20 or so dead ones. The bunt pans are different. This is one sampling. So the bunt pan is over here on the left. You can see that down below. In that case, we're getting more dead ones than alive. Now, I don't know. It's hard to say whether that's a function of the fact that they've gone into the bunt pan and die soon after because of they're not in soils. But regardless, if we put those numbers together, they are lower even when we combine live and dead than what we see even just alive in the soil and glacier deposits. And then OTC is an open top chamber, and I'll show you a picture of one of those here in a second. But we find a lot of dead organisms in the open top chamber samplers, and not too many live ones. Now, on the left here, you can see Diana Wall, one of the founding PIs of this project. This was a wetting experiment. The open top chambers that were sampled for this graph that I'm showing you were not treated in any way, but some of them are warmed. Some of them were meant to have added water or nutrients. And so in this case, what we're seeing is that when we just compare adults and juveniles, which may give us some indication of how much proliferation of these organisms is occurring. We see quite a few juveniles in the soil and the lake and glacier deposits, about even in the bunt pans, and then a lot more juveniles than adults in the open top chambers, which is sort of interesting. Now, they don't move that very far on their own, so it's not as if they're suddenly collecting here for some reason. But we do expect this is sort of interesting that there are so many juveniles in the open top chamber capture compared to the adults. Well, you won't be surprised to know that, of course, sublimation and melt occurs and evaporation of our open water occurs in such a dry location as well, despite the fact that it's really cold. Now, we can think about these ice covers, and one of the things that we've done is tried to monitor the water level in lakes at a very high frequency. And during the winter, this is fairly easy. The ice builds up. We can still measure a static pressure underneath and figure out how much liquid water is being converted to ice, and therefore, what's happening to the water level underneath. And some of that is also reflected in, if we account for the mass of ice in the ice covers, that any that is removed is also figure that out as well through our every 15-minute data collections under the lake ice. And then during the summer, of course, we have meltwater coming in, and we've got open water around the edges, so we can have evaporation occurring. And so when we put all this together, the two plots down here, this is 3 years of this lake, elevation data in 2 different lakes. So Lake Hoare on top and Lake Frixell on the bottom. And you'll see that the big step ups occur in the summer. So this is the net gain of water from everything that's coming in, but we've also accounted for the loss that's occurred there, right? So whether it's evaporation or sublimation. And then during the winter, we see this steady decline at a lower, overall magnitude than the step up that we have in the summer because of the sublimation of the lake ice. So when we put this together, we estimate 7 to 21 millimeters of water loss in the winter due to sublimation, and in summer about 25 to 162 loss of water, which is that combination of both evaporation and sublimation of the ice covers. So connection to the atmosphere, obviously very important, when particularly in the case where we don't have a ton of snow being put in. So we'll just kind of wrap this section on wind and just noting that these phone winds are mostly occur in the winter, so there is connectivity going on when we're not there. We tend to see the results of that, and we can go sample it. We see lots of material that's been moved around. That includes nutrients and organisms, and of course there's this connection to the atmosphere where we're losing water, whether it's ice or liquid water from a number of these places. All right, let me switch gears here and talk about streams for just a little bit. This is something that I study more closely, and these streams are fascinating. I mean, they do connect glaciers. Most of the melt or the liquid water comes from glacial ice melt. And they flow for about 6 to 12 weeks per year. Sort of depends on the stream, depends on the glacier, which way it's facing, how dirty it is, that kind of thing. And some of them they're all intermittent in the sense they don't flow all year, but some of them, go for 6 to 12 weeks and don't stop. And then some of them are kind of intermittent. They'll go for a couple of weeks and then slow down with a storm that comes through, et cetera. And so there is some in-season intermittency that we qualify as well. Now, we have about 17 stream gauges set up throughout the dry valleys, and they may look something like this on the left. There is typically one of these large crates, as we see on the right here, where we keep all of our instrumentation and batteries and so forth out of the elements. And then we also have a bubbler system, so we have nitrogen tanks in there to bubble nitrogen out to measure the pressure of water, the height of water at each of these sites. And we record discharge really staged. We convert to discharge after making manual measurements. Water temperature, electrical conductivity as a surrogate for salinity, and then dissolved oxygen is a new one that we've started just a couple years ago to try to back out metabolism of these streams. So how much primary productivity is going on in the stream water and how much respiration is occurring as well. We'll just stick with looking at sort of the salinity and flow. And this is the 2008-2009 flow season for 2 different streams. Canada Stream is a very short stream. It's about kilometer and a half long, and Delta Stream is about 11 kilometers long. So these are 2 very different types of streams. And you'll notice that the short stream, the flow lasts longer. There's a longer flow season, but they both have this characteristic sort of high frequency pulsing to them. And that really is a daily pulse. I'll show you that on the next slide. We'll zoom in on 1 week for both of these. The other thing to note is in red, you'll see that we've got the electrical conductivity plotted, and it's typical to have fairly high electrical conductivities, or rather higher salinity. I mean, these are not super high. We're not getting more than 400 microsiemens here, but then we kind of dilute out in the middle of the season when flows are high, and then kind of ramp back up towards the end of the season. That's muted in Canada Stream, you can see, and in Delta Stream, it's much more pronounced. Now, the other thing I want to point out here is that these long streams tend to have higher conductivities than the short streams, and that's partly because that water is interacting with a lot more sediment. We'll go into that detail here in just a moment. If we zoom in on just a week or so of these data, you can see that daily flood pulse. And this is just from the sun circling the sky. Even though it doesn't go down, it still, is intense on a glacier face for a while, and then it keeps moving. And so you can see these pulses, and we try to capture different, aspects of this, whether it's going out and trying to get high flows or low flows each day and trying to make sure that we're capturing this dynamic. Now, the Delta Stream record is a little rougher than the Canada Stream one, and the reason for that is that it has a different gauge layout, so it's not as smooth. Canada has a very nice flume built into it. Delta is more what we call more of a natural control. It's a bunch of rocks with some tarp over it to funnel the water through one location, but it can easily be susceptible to wind compared to Canada Stream. Okay, back to our map here. I'm going to talk about the Onyx River for the next slide. And I just want to point out that the Onyx River is in Wright Valley, so this upper right plot. And north is more or less up on this particular map, but the coast is far off to the right. This is actually a Piedmont Glacier, and then at the edge of that Piedmont Glacier is Lake Brownworth, and Lake Brownworth yields water that feeds the Onyx River. That flows about 30 kilometers inland to Lake Vanda. So the terminus of the Onyx River is Lake Vanda. And we have a stream gauge up here called Lower Wright Glacier. And so LWRT is what you'll see on the next slide. And then right before the Vanda, before the river enters Lake Vanda, we have another gauge there. So I'll just show you these data that go all the way back to the 1969, I believe. So the 1970 season here. We have both Vanda and Lower Wright. So the Lower Wright is in sort of this light blue color, and the dark blue is Lake Vanda. And I'll just note, or not Lake Vanda, this is the Vanda gauge. But what's really interesting here to me, has been super interesting, is that often there's more water coming through Lower Wright than going through Vanda. So where did it go? And it's probably coming back. I've tried to estimate transmission losses of water between these two gauges in the past, and I haven't done a very good job. So I'm not exactly sure I can pinpoint, except to say it's evaporation to some extent. And probably also when the bed sediments thaw out, there is a big sponge that has to be kind of filled in with water before water can keep moving downstream, right? It's like you've got to satisfy that volume first. But the Kiwi program ran these stream gauges from their inception. They put them in place and ran them through about the early 90s. They handed it over to us, and they still worked the records until about 2000, and then we took over the record keeping as well. So this is a really interesting collaborative way of sort of thinking about these, the way in which we collect data out there. And I'll just note two things real quick. The numbers here are real in it was this one, 1977. There was very little flow that passed Lower Wright, and none of it made it down to Lake Vanda. So that was kind of a fascinating year. But on the other end of the spectrum, we had a flood year in 2001, 2002 austral summer that really started in about mid-December, and that'll become important here in a second. Now, these streams and rivers mostly flow to closed basin lakes. And so the closed basin lakes are a record of climate to some extent, which is to say that if they are rising, then they are receiving more liquid water from meltwater and losing less than if they were staying stable or dropping, right? And so here's what we see since 1972. So zero is just 1972 is when the Kiwis actually were out making lake level measurements at a number of these lakes. So these are the three main lakes in Taylor Valley. So we've got Bonnie in blue, and that's the high one here. And then the red is Frixel, and the green is Lake Hoare. And they generally move in concert with each other, right? Hoare and Frixel, they've only started to separate recently. And then Lake Bonnie has had a much greater rise. And part of it is the hypsometry of the valley. It's a little tighter of a location right there. But almost 9 meters rise since 1972, and for Lake Bonnie, and about 3 or so at both Frixel and Hoare. Now, right in here, you can see the flood year when it occurred, this big step right here. And that was the year that we also published, I wasn't part of the paper, but we published this paper about Antarctic ecosystems responding to decadal cooling, which was of course picked up in a variety of misconstrued ways through the popular press. But you can see that there was about a decade of sort of negative mass balance in these lakes. And then we had the flood year. So if you want things to change in your system, write a paper, publish it, and within that month, when you publish it in Nature, the system will go wacky and do something different. So that's what we learned. But there's also this hypereic exchange. So there's a connection between the the streams and their beds. And so I talked a little bit earlier about filling up that sponge underneath, but there's active exchange that's occurring whenever stream flow is moving from upstream to downstream. And we call this the hyporheic zone. Hyporheic is just identifying the fact that there's water that's moving from the channel into the bed and then back into the channel again. And the consequence of that, in this case, is a lot of weathering of that material, right? This is a very dry landscape. I didn't mention it earlier, but we have no rain here. We do get this snow, but a lot of that sublimates. We don't get a ton of snow melt here moving water across the valley. So the sediment that gets a lot of exposure to water is in the stream bed in this case. All right. So we have really high weathering rates. This was a modeling study that I conducted for my PhD work. And at the time, these were the highest silicate weathering rates that we could find in the world. And that might really surprise a lot of people from the context of saying, well, this is just, it's so cold. How could you have this? Well, the fact that it's so dry and these sediments don't see a lot of water means they're fairly fresh. They are ready to weather. And secondly, they're pretty simple. I mean, they're just kind of hanging out there, waiting for water to come through, and then they only see it maybe 6 to 10 weeks a year. So this is very different than the context of Coweta or Loch Vale or Bear Brook, where I just compared these different sites to our silicate weathering in the dry valleys. We also, I mentioned earlier that we see diatoms in these stream beds, or sorry, in the mats on the streams. And this plot on the right, I just want to note, don't feel like you have to understand this completely. Let me walk you through it. It's a dot blot plot, which is indicating in this case, on the left, we see samples collected from the left y-axis, samples collected from different streams during different periods of that stream being turned on or turned off, different seasons. And then across the top, we have different diatom taxa. So one sample that's been processed would be identified on the left y-axis, and as you go across, the contribution to the total community that's there of diatoms is represented by the size of the dot. Okay, and so what's interesting to look at is that the y-axis has been organized from the most consistent streams to the least consistent streams. And so you can also see the shift in the size of the blots, right? As you move across, you can see that the taxa that are indicated on the left are more common in consistent flowing streams than the ones at the right. And those are more common or represent more of the diatom community in the inconsistent flowing stream. So this is one way of just sort of indicating that we see these shifts. Diatoms are often used for thinking about water quality, but we see this almost as a season population and dynamic. So the streams connect these ecosystems. They connect the glaciers to the lakes. They provide habitat for diatoms and algae, and the biological communities. These diatoms are very sensitive to flow, and the streams are not mere pipes, right? There's this interaction between the stream flow and the bed underneath to drive this weathering signal and influence the chemistry. All right, my last section here, I'll try to move through it quickly so we have some time for questions, but we often have been thinking a lot about what happens from one summer to the next, which is to say, what happens in winter. We all have to leave the field by early to mid-February, and we don't really know what's going on. We can get back in the field if things are going well in October the following year. So how do these ecosystems survive the winter? Now, in a lot of cases, when we're talking about the soils or the streams, by and large, they are going into some kind of desiccated mode. They're not active. But the lakes have an ice cover that might be 3 to 5 meters thick, and then they've got liquid water underneath. So there are opportunities. Those organisms have to somehow make it through the winter to survive and be present the next year. So 3 to 5 meters means that if you're going to drill a hole, you got to drill a really long hole. So not uncommon for us to use jiffy drills that take many flights to put together to go through the lake ice. And then sometimes we melt out a larger hole. And so in this case, we will drill these holes, melt them out, and then get a hut built on top of it by the carpenter crew in McMurdo, and then do our sampling inside of this little hut. So there's a little polar haven out there. And you can see on the right, Amy here is sampling within. So she's looks like she's dropping a CTD to do a cast and sort of get a profile of temperature and conductivity and pressure and so forth through these systems. So so we've got a number of oh, actually that's a sampler. Now I see, I was looking down on the hole, but in any event, this is what we do sort of 3 times a year on each of these lakes. But the lakes are connected to the streams, and the streams are connected to the watershed. And so when we had this big flood event in 2001, 2002, you can see that the lakes responded. So in this case, primary productivity is by the phytoplankton in the lakes is generally occurring. I believe this is Lake Frixell at about 13 meters depth is kind of our peak where just enough light comes through and there's a bunch of nutrients coming up from underneath to meet right here. And in November 9th and the 4th of December, high peaks there. But the 28th of December, after this flood had started, there was a whole bunch of sediment in the suspended sediment in the lake, and it was affecting how much light we get through. And so we see that the primary productivity crashed on the 28th of December. You can see that there was very little peak here, and it was pretty low all the way through the water column, which is fascinating to see, but it's indicative of these connections. We've been able to get a few other grants. This one came from a NASA grant, I believe, to study what's going on in these lake water columns through the winter. Now, we have a phytoplankton sampler, this remote access sampler, we had a profiler moving up and down, and then we had a sediment trap that stayed down at the bottom. These two ended up, this ended up being a fixed depth, and this profiler moving up and down was actually would only go a couple of meters. We thought we had the design worked out for it to move the whole way, but the power problem became an issue in the middle of the winter. And so this is what these systems look like, right? You've got to melt a pretty big hole to get these in, but one of them will collect samples. Another one will, when it's filtering those samples, it will collect the filter and save it. So we've got just a beautiful suite of data to look at. We also have a flora probe out there and so forth. And one of the things that we see from this is that the chlorophyll A concentrations tend to peak in August, right? And this is, I want to say these are mid depth. So this is right around sort of 10 meters depth, something like that. And there is some standard deviation on these measurements, but this peak in August is pretty fascinating, right? It's been dark for a couple of months. Why are we peaking with our biomass then? It has something to do with the nutrients, even though there's not a ton of light. If we break that algae down into a couple of different types, we've got chlorophytes, cyanophytes, chrysophytes, and I'll just note that this crash in the chlorophytes is not necessarily, they haven't gone away altogether. It's just they've gone away from this section of the lake. And we see at that time the chrysophytes taking off then. So this is an opportunity to think about how we connect from one summer season to the next, at least in these lakes. Finally, I'll just give you one kind of cool physical example too. Zooming in on the west lobe of Lake Bonny and the east lobe of Lake Bonny, Scott's men came through in 1903 and they measured the width of what we call the narrows here. And we've been trying to kind of figure out what the lake level was, because the lake level has risen quite a bit since 1903. And we've had a number of dive teams working there at this particular location, but we've also figured out that these two lake basins are different. So the east lobe of Bonny on the right-hand side here has this black profile here in salinity, so much saltier, and peaks at about 28 meters depth, something like that. The west lobe peaks much higher, and much shallower, I guess I should say, and then doesn't get quite as salty. And so one of the things that we've been thinking about is what goes on at this pinch point, where we've got the narrows occurring here. Are there flows of water moving from left to right, moving from west lobe to east lobe? So if we line these up, we see a little bit of a notch there, and that kind of lines up with where this is just the conductivity profiles. And sure enough, for the most part, there's a little notch there, which suggests there's some kind of communication. And so we put a conductivity probe there, and through time, you see these spikes, right? Those spikes we are suggesting are burps of water moving from one side to the other, moving deep water coming from west lobe, moving into east lobe. And the reason this is most likely is that on the western edge of the west lobe is the Taylor Glacier, and the Taylor Glacier extends out to the rest of the East Antarctic ice sheet. So if it's moving into the lake a little bit, pushing some of that water over, it'd be interesting to know how these are connected. I will say we work with Antarctic New Zealand a whole lot. This is Ian Hawes. We've been working with him for a number of decades now. This is the setup that he helped place at this location in the Narrows to capture these data. And we have that better instrumented now, most recently, about 2 years ago. The other thing that's happening is we're starting to see a lot more atmospheric rivers. And so in March of 22, we had a peak air temperature that went above 0, and it was 7.1 degrees higher than our 30-year average at this location. And we even saw some liquid water, or at least wet spots occurring in one of our stream areas. So you can, we used MDVI to sort of indicate that we were seeing the wetting of algal mats that were there. So this connection to the broader climate cycle is important as well. All right, I'll wrap up here. Apologize, I haven't given a whole lot of time for questions, but the dry valleys are as interesting now as they were in 1903. Certainly when they were discovered, that was fascinating and important and interesting. But you know, we now think about it in the context of, you know, they thought of this as the valley of death. They didn't see the evidence of life, and we see it everywhere now. And it's, I mean, granted, if you find you know, mummified seals, that suggests there was previous life. But with our new approaches to thinking about how these systems work, we see it differently. And we're now trying to really understand all these connections that drive this ecosystem. It's certainly a privilege, and we feel like a responsibility to work there. We need to publish our data. We need to, you know, mentor awesome scientists that move on in the world and do great things. It's a barren landscape that really has, you know, life that doesn't think of it as stark or as you know, inhospitable the way we do as humans. We just have different tolerances. Many of our discoveries come from a lot of patience, a lot of persistence, and a lot of careful planning that starts, you know, often the year before, if not many years before. There's actually a stream under this graduate student. She was just collecting a sample there on top of this glacier. And, you know, these are the things that we can do there and try to better understand glaciers broadly, not only in the context of the dry valleys, but other places as well. And so we keep studying the dry valleys. We want to figure out how life works here and study the responses to the changes as they come. You know, it really has been an honor to work here for so long, and we're looking forward to another field season coming up, starting in just next month, actually in October. We'll have folks deploying. So finally, let me just acknowledge, I gave you that little snippet of a video earlier. Ariel Waldman has this really cool movie called Antarctica Unearthed that she's trying to get distribution for right now. It's about an hour long. If it's coming to your you know, somewhere near you, or with a showing, or you find it on Netflix or something, do take the time to look at it. It's pretty interesting. It doesn't really go into our story at all. It's really just about this landscape at multiple different scales, including the microscopic, the life one. Finally, I'll just acknowledge that the work that we do out in the field, obviously, it really comes from a lot of different places in McMurdo. There are probably 8 contractors to every single scientist that gets to deploy in the field. And so I want to acknowledge, I'm not going to go through all these acronyms, but you know, it takes a village to get our science right, so to speak. And I want to make sure that we don't ignore that. And then finally, I'll just note that we have some social media. So if you want to check us out, we do have this MCM_LTER on Instagram, and you can keep up with some of our comings and goings when we get to those. But with that, I'll wrap up, and appreciate your attention and happy to answer any questions as time allows.
Thank you, Professor. Thank you for taking your time and sharing with us your expertise. So for the online attendees, if you have any questions, you can chat us your question, or we encourage you to speak up. So looks like we have a question in the chat. So Santiago, do you want to go ahead?
Well, I mean, it was partially answered, but I guess if you could expand a little bit about how these lakes are. I mean, they seem, yeah, I saw there that some of them can be tens of meters deep, but I was curious if they are permanently or they can be frozen all the way down through sometime during the year, or is water permanently there?
Yeah, thanks. I didn't go into the different depths. Some of them are only 20 meters deep, and some are 70 meters deep. So the most of them do not freeze all the way. And part of the reason for that is not only the thermal mass that's there and what's happening at the surface, but they're also hypersaline down at the bottom. Most of them, a few of them are not. But at the bottom of Lake Bonny, for example, you can actually find crystals, salt crystals that have formed because of the pressure and concentration, and pull them up, and they're not very stable at the surface when you get a quick picture before they disintegrate. But there is one lake that's has a very thick ice cover, and that's Lake Vida, which is in a Victoria Valley that I didn't talk about much. It's a little bit north. It tends to be a cold place, but I think if I recall, it's about 20 meters of ice and maybe 6 meters of water under that. So very different dynamic there. But yeah, thanks. Great question.
Any other questions?
I was anxious to defer to my colleague, Byron Adams here, who joined us.
Oh, that's great.
Well, I
So-
mean, can I ask again? This is Santiago. I mean, I'm fascinated by the subject in general. My understanding of this subject is coming from the trying to understand how dust is produced or generated in high latitudes. So I've been doing that more from the remote sensing side and looking at dust blowing in Alaska and Greenland. But then I did come across some papers reporting dust blowouts in the dry valley. So that's why I was curious about this talk. Yeah, but I mean, you mentioned is there a good understanding of the ecosystem and the lakes and what limiting factors are with respect to the nutrients? Is there a good understanding of that, or the biogeochemical cycle?
Oh yeah, there is. I would refer you to a number of John Priskew's papers for that. But I'll just as a quick snapshot. So I mentioned that the lake bottoms were generally hypersaline. They also have really high concentrations of nitrate at the bottom of the lakes in general. So there's this sort of diffusion up through the water column, which is obviously pretty stable. And that's why we see that peak in primary productivity and chlorophyll kind of deep, like right above the chemocline, because that's where you get a sweet spot between the light coming in and the nutrients from underneath. But yeah, he's got that pretty well organized if you wanted to look through his papers from the 90s in particular. Yeah.
Fascinating. Yeah. Have you found connections with what you find here and in other similar places in the Northern Hemisphere, Greenland, or are there any similarities you can comment on?
Yeah, I'm not sure I could speak for the lake context, but after I finished my PhD work here in the Dry Valleys, one of my first NSF awards was to work up at Toolik Lake in Alaska. So I started thinking about, okay, what happens to those kind of streams? Like they're still underlain by permafrost, but what happens with this hyporheic exchange, for example, and all this carbon around and so forth, and rain, right? And we've got rain that occurs up there. And it was, they're similar and different, like as you'd expect. But one of the similarities is just that you've got limited interaction with respect to the bed and not an aquifer, big aquifer that you're connected from the stream perspective. And so that has some important ramifications, but you do see that like some of those beds stay thawed in the winter because you have a couple meters of water and ice above them. And so, that would allow for biogeochemical cycling of whatever is there available in those sediments all the way through the winter and then starting up again the next spring, right? So they're kind of it kind of in the Arctic, it sort of depends on where you are. Like a beaded stream kind of fits that description I just gave you, but an alluvial stream may not. It may freeze all the way to the bed. Yeah. So
well, thank you.
Sure.
Thank you. So any other questions?
I see Frank's question there. So so great question. So and dang it, Byron got off just before I could have asked him that question. So Byron does a lot of our worm counts. And so we bring the samples back and go through a process of processing the sample and then get them onto a slide. And and then do counts underneath that slide. And so there is a scaling approach to this, right? Like we don't it's maybe 100 grams that was the sample size. We can count the number of organisms. We have to scale that to a kilogram, but he's gotten pretty good at figuring out which tardigrades, for example, are alive or dead. And I think I have not gotten to actually do these counts myself, but my understanding is that they are wetted first, so that and sort of at room temperature, so they shouldn't necessarily be in that cryptobiotic state anymore. But a fair question to ask in the context of just thinking about what state they're in when we capture them from the field, so to speak, in our sampling, and then count them in the lab. One thing I will say is there are a couple of measurements and analyses that we want to do in Antarctica because we don't trust them to be the same if we ship samples back. And one of them is live dead counts, for example, in our soil samples. And the other one is analyzing dissolved organic carbon in our liquid water samples. So whether those come from the lakes or the streams, we don't expect that dissolved organic carbon to be the same character and amount necessarily by the time we got it back to the US. So those analyses are done in McMurdo before we leave. So thank you.
So I think any other questions? If not, we are going to conclude the seminar. I want to thank the speaker again, and I want to thank everyone for showing up. I hope you have a good rest of your day. Thanks.
Thank you very much.
Thank you.
You bet. Take care.
Bye-bye.
Bye-bye.
Bye-bye.